Hi, my name is Gabriela. I'm a postgrad researcher at the Center for Psychedelic Research. And in my presentation, I will try to answer the question of how studying brain dynamics can help us understand the effects of psychedelics. I'm sorry. Okay, so what is a dynamical system? In order to answer this question, let's deconstruct the term dynamical system into its components. So the word dynamics simply refers to a way system changes or behaves as time passes, and this also applies to us humans, as you can see on this picture. A system is some collection of related parts that we perceive as single entity. And for example, the systems we are all familiar with are the solar system, the capitalist system, the nervous system, or the telephone system. A dynamical system is one with changes in time, and what changes is the state of the system. And the consideration of time or dynamics is fundamental for all aspects of mental activity, such as perception, cognition, and emotion, because the main feature of brain activity is the continuous change of the underlying brain states, and it occurs even in a constant environment. In terms of early views on brain dynamics, Erwin Schrödinger and his work on thermodynamics had an important role on our understanding of the essential forces of life. And in his book, What is Life? He argued that for a living organism to exist, it must never cease to contribute to the total entropy of the universe. And entropy is a measure of the level of disorder in a system. So according to his views, the ultimate equilibrium is death, and thus survival depends on staying as far as possible from this equilibrium. Um, Practical applications of dynamics to the study of brain activity began in the middle of past century, but such research only flourished when combined with some empirical observations of brain activity from modern morphological and physiological experiments. And this new perspective was celebrated in a book, Mind as Motion. And this book reflected the changing views of neuroscientists, psychiatrists, and others trying to understand human cognition, mood, and emotion. And this book promoted a shift of emphasis from localizationism, which postulates that brain is functionally segregated, that parts of the brain and not the whole perform specific function, to studying the brain activity in time. Application of dynamics to the study of brain activity gave birth to various frameworks and theories. And I'd like to present two of the most recent ones, which have been effectively used to enhance our understanding of the effects psychedelics have on the brain. And these are namely the Dynamical Connectome framework and the Connectome Harmonic framework. So let's start with the Dynamical Connectome framework. Connectomics is the study of brain structural connections between cells, which is visualized as a connectome shown here in this figure. And human connectomics has been very successful in revealing how function arises from structure, and by showing how anatomy can give rise to a complex dynamical neuronal system. However, despite the attractiveness of the idea that function is shaped by anatomy, it is also clear that the fixed structure of the human connectome should not be able to give rise to the full palette and complexity of brain function. And therefore, one of the most exciting challenges in the field of modern neuroscience is to understand the dynamical complexity of the human brain. And specifically how mental states arise through this complex interplay of structural and functional brain connectivity. And this has been demonstrated in this study, in which a theoretical framework, the dynamical Connectome framework, has been developed to model the dynamical mutual coupling between the neuronal system and the neurotransmitter system to explain the functional effects of specific serotonin receptor stimulation with psilocybin in healthy humans in a resting state fMRI trial. And this is an overview of the coupled neuronal neurotransmitter whole brain model. And it has been built by adding a coupled neurotransmitter system, which is here in blue, to modulate and interact with the neuronal system here in green. And this neurotransmitter system was modeled using a special mathematical model that expresses the time evolution of the collective activity of different neural populations, which are collections of excitatory and inhibitory neurons. And this model was then simulated to investigate the role of specific parts of the dynamic coupling. And specifically, the results show how underlying dynamics of they provide insights into the underlying dynamics of neurotransmission involved in psilocybin. And the data in the graphs are expressed as KLD for PMS, which describes how much the simulated spatial temporal dynamics differ from the corresponding empirical dynamics. So in terms of dynamics, this first figure shows that uncoupling of the neuromodulator system, which describes the dynamics of the neurotransmitter concentration levels from the neuronal system, produces a highly significant breakdown in fitting the empirical data. In the next figure, the full mutually coupled neurotransmitter neuronal model ran until a steady state was achieved, upon which they froze the feedback dynamics from the neurotransmitter system to the neuronal system by simply removing coupling for the raphe nucleus, which is the main source of serotonin in the brain. And this again produced a highly significant breakdown in fitting the empirical data. In further sets of experiments, which were designed to investigate the importance of serotonin receptor distribution, the regional distribution of the specific serotonin receptor 5-HT2A receptor has been randomly shuffled. And this figure shows the significant difference between using the empirical 5-HT receptor densities compared with the results of randomly shuffling the receptor densities. And collectively, these results show that by integrating the information on anatomy, functional neuronal activity, and neurotransmission at the whole brain level, we are able to get a more holistic insight into brain function under psychedelics. And that in silico simulation of these systems allows us not only to study the tight interactions between the dual neuronal and neuromodulator system, but also to properly predict the behavior of that brain dynamics upon the administration of psychedelics. And why is the study so important for future? So overall, the dynamical Connectome framework is likely to be essential for accurately modeling and explaining mechanisms of human brain function in health and disease. Therefore, it could be used in a novel branch of personalized medicine approach, in which modeling of the dynamic changes in neurotransmitter concentration and neuronal activity could be used to make predictions on how to rebalance human brain activity and disease through, for example, pharmacological interventions, through electromagnetic stimulation like deep brain stimulation or transcranial magnetic stimulation. And in the future, this framework could be further extended by measuring even faster whole brain dynamics through magnetoencephalography, or incorporating gene expression at the whole brain level, which offer even more precise avenues for discovering newer and even more effective ways of rebalancing the human brain in disease. And the next framework I'm going to describe is the Connectome Harmonic framework. I want to first define the brain state. So a brain state is an elementary state of brain activity. So just like different combinations of musical notes create infinitely many different pieces of music, it may be as simple as a lullaby or as complex as a symphony. Different combinations of elementary brain states can also give rise to an infinite number of possible variations of spatial temporal patterns of brain activity, which link space and time. And the definition of brain states as the connectome harmonics links space and time in brain activity via the universal principle of harmonic waves. And harmonic waves are ubiquitous in nature. They emerge as building blocks of pattern formation in various physical and biological phenomena. For example, the standing wave pattern of emerging in a sound-induced vibration of a guitar string is an example of that. And the Connectome harmonic framework, it provided a novel method to decompose the resting state, fMRI measured cortical activity pattern under LSD and placebo, into a set of independent and frequency specific brain states, the harmonic brain states. And in a way, translating this fMRI data into this harmonic language is actually not very different than decomposing as a complex musical piece into its musical notes. So this is an illustration of the workflow. First, magnetic resonance imaging data was used to reconstruct the cortical surface between gray and white matter, and diffusion tensor imaging data was used to extract the thalamocortical fibers. Then both local and long distance connections were combined to create the connectivity matrix of the connectome. And at this stage, connectome harmonics were computed via something called the eigendecomposition of the Laplace operator, which lies at the heart of theories of heat, light, sound, gravitation, and fluid mechanics. In the next stage, the fMRI imaging data was decomposed into the activation of connectome harmonics, yielding the power of activation of each of these brain states for each time instance. And the main aim of this study was to test whether LSD induced any changes in the energy and power of different frequency connectome harmonics, which are the two fundamental properties of harmonic brain states. And through evaluation of power and energy of harmonic brain states, the sensitivity of cortical dynamics to the effects of LSD can be estimated. And the primary analysis revealed that the total power and total energy of all brain states averaged across all time points significantly increases under LSD between each pair of LSD versus placebo conditions. And further analysis investigated which brain states demonstrate the increased activity under LSD. And to do that, the connectome harmonic spectrum was decomposed, was categorized into 15 different levels of wave numbers. And then the energy changes within each of these parts of the harmonic spectrum were analyzed for each of the 6 conditions. And the results show that LSD increases the energy of high frequency brain states, and it actually decreases the energy of low frequency brain states. And this means that LSD alters the energy and the power of individual harmonic brain states in a frequency selective manner. It also expands the repertoire of active brain states by significantly increasing the activity of high frequency brain states. And then to further characterize the energy spectrum of each of the six conditions, the probability distribution of their energy values were estimated. And there was a clear shift to higher energy values at the peak of probability distribution in all three LSD conditions in comparison to the energy probability distribution of the corresponding placebo conditions. And moreover, in the LSD with music condition, the probability of reaching the maximum energy state was higher than the initial LSD condition. And these results suggest that LSD causes cortical dynamics to change more rapidly with increased flexibility, which may potentially underlie the enhanced sensitivity to the environment and context, which are observed under the influence of LSD. And these dynamical changes of cortical activity were then explored in further detail in criticality analysis. And although I don't have time to go through it today, I will show what these results can tell us about the potential use of LSD in therapy. So we know that LSD alters the energy and the power of individual harmonic brain states in a frequency selective manner, and it enriches the Connectome harmonic repertoire. And this actually indicates that more brain states contribute to neural activity under LSD, leading to a higher and more flexible repertoire of dynamics. And this effect is an expected property of brain dynamics approaching something called criticality, which is the property of being optimally poised between order and disorder. So it's like a state that is maximally diverse and flexible while retaining the properties of order. And therefore, it has been hypothesized that this increased energy and enriched repertoire of brain states under LSD could coincide with tuning towards criticality. And this hypothesis was later confirmed, and this finding has several important implications. So firstly, criticality provides necessary conditions for optimal information processing and representation for a complex system, making it more flexible within its own intrinsic functioning, but also more sensitive to incoming stimuli. And secondly, various previous studies have shown that deviations from criticality could be symptomatic or even causative of certain psychiatric disorders. In particular, brain dynamics in depression, addiction, and OCD have been associated with subcritical regime. Whereas supercritical regime has been found to govern brain dynamics during epileptic seizures and in conditions such as autism. And taken together, these findings highlight the potential effects of LSD to regulate brain dynamics in pathology by reestablishing this critical balance between ordered and disordered states. And this may therefore this may therefore explain the increasing body of evidence supporting the therapeutic potential of psychedelic drugs for treating disorders such as OCD, depression, and addiction. And maybe actually a phenomenon that stands behind the richness of consciousness under psychedelics, and the notion that they expand consciousness. And this is the list of references that I used. I would like to thank you for listening to my presentation, and I want to thank Ellen, Bianca, and Craig for their support and stimulating conversations we've had during our team's meeting. Thank you.